We have Kanmani Chandra Rajan joining us. She is a molecular biologist from Tamil Nadu, one of the southernmost states in India. Currently, she is working as a postdoctoral researcher at the School of Biological Sciences and the Swire Institute of Marine Science at the University of Hong Kong. Her current research is ocean acidification and invertebrate biomineralization. She also makes Tamil science communication videos on her YouTube channel, The Tamil Scientist. So we're going to share a link to that shortly. But for now, let's bring Kanmani in with us. Hey, how are you doing today?
I'm doing good. How are you?
Good, good. And I believe you're in Hong Kong today.
Yeah, I'm in Hong Kong.
All right. Very, very cool. Well, it's so great to have you with us. I'm a little biased. I love the ocean. I love diving and exploring the ocean. So anything that talks a little bit about our oceans is right up my alley. So I'm really looking forward to your presentation.
Thank you. Thank you so much for this opportunity, Joe. Thanks for contacting me and giving me this opportunity. I'm so glad I got this stage. Thank you.
Of course. Well, let's let you take over for a little bit. We'll let you run the show and then we'll ask you a few questions.
Yeah, sure.
All right. So we saw that screen share practice backstage and it looked pretty darn good. I think you're a pro with the screen share.
Yeah, let me try now. I hope
Yeah, absolutely. We've had an adventurous day today where sometimes technology has been flawless and sometimes we've had to wrestle a little bit, but I see your presentation. It looks nice and full screen and we'll leave it to you for a bit.
Yeah, sure. Hi, everyone. Thank you for joining my talk today. I am Kanmani Chandra Rajan, and today I will be talking about Hong Kong oysters, in particular, Hong Kong oyster shells and how they perform under ocean acidification. And before starting the talk, I would like you to look at these oysters. So you have oysters in the shell, there's meat, and I would like you to particularly focus on this dark part in the meat. This tissue is called as mantle tissue in the oyster, and this tissue is important for forming shells in oysters. So I'd like you to remember that first, and let's get started now. So today I'll be talking about Hong Kong oysters, a particular species in Hong Kong, which is called Chrysostria hongkongensis, which is recently renamed as Magellana hongkongensis. And I will talk in particular about oyster shells, Hong Kong oyster shells, and the shell formation process called as biomineralization, and how the oyster shell formation is affected by ocean acidification. And I will also finish the talk with our effort in building Hong Kong's first oyster hatchery. So our research group is building Hong Kong's first oyster hatchery with the help of Hong Kong government. So I'll talk about that, why we need a hatchery and what we are doing. So when everyone think about Hong Kong, this is the picture that comes to most people mind. It's like the Hong Kong is famous for its skyscrapers, nightlife, and all the party. But Hong Kong is also has a cultural heritage for oyster aquaculture. Over 700 years of cultural heritage in oyster aquaculture. In this picture, you can see oyster wraps floating in the ocean surface, where these wraps in the from the raft they hang the oysters down into the water, and the oysters feed the water, and they grow in these racks. And then after a year or two, the farmers cultivate these oysters, they harvest these oysters, and they sell it in the market. Hong Kong alone produces this particular species 100 tons of meat alone annually without the shell, according to the Agriculture and Fisheries Conservation Department, Hong Kong. So 1-year-old oyster can filter up to 30 liters per hour of water. This is very high comparing other oyster species, which many species they do around 3 to 4 liters, but this particular species is very efficient in filtering waters. It filters up to 30 liters. And also the another special feature of this oyster is its shell is very strong. Comparing Portuguese oysters, this particular Hong Kong oyster can withstand much higher crushing force. So when you try to break the shell, it can withstand much higher crushing force. So their shell is much stronger than the related sister species. So I am particularly interested in the shell and shell formation and how it's affected by ocean acidification. And before getting into ocean acidification and shell formation, I would like to introduce oyster life cycle. So oysters, this is the typical life cycle of oysters. This cartoon shows how adults they release eggs and sperm into the water column. Oysters are sequential hermaphrodites, which means they can change their sex from they can be male and also female after some point of time. Like for 1 year, they are male in one season, and then after that, they can become female. So they release eggs and sperm into the water column, and after that, the eggs and sperm fertilize, and they form small larvae, D-shaped larvae, trochophore, veliger, and pediveliger are various stages of larvae. And then after they reach this pediveliger stage, they are ready to settle down. So the larval stage is free swimming. They can swim in the water. And then after that, they attach to a hard surface like rock or already existing oyster shells in the habitat. So they settle down and continue to grow as adults. So this is the life cycle of oysters. So now we go into shell formation, or the process called shell formation is called as biomineralization. So when we all look at oysters, we see this shell, right? So this is what is visible to our naked eye. Those shells are beautiful, even just with naked eye. They have even more beautiful structures in there. When you look at them under microscopes, particularly electron microscopes, they can reveal so many beautiful patterns. In this picture, you're seeing one particular pattern, folia structure. So this structure is called a Scolia structure. It's one of the typical structures found in Hong Kong oysters. And for forming these different patterns, different structures, oysters use this biomineralization process. So as I said, the black color tissue, I asked you to remember in the beginning, the mantle tissue. This tissue is known for secreting proteins that capture calcium and carbonate from seawater, and that fabricates the shell. And for making different patterns of shells, you need different combination of these proteins. These proteins also serve as a template for forming the different shell microstructures. So this is how shell is formed. And also for this process of shell formation to happen, there is requirement for calcium and carbonate ions from the seawater, and also there is a requirement for pH control at the site of mineralization. So this oyster has to control the pH at the site where it's forming its shell. So pH is also very important for this process. So ocean acidification is known to be negatively impacting several marine calcifiers, including oysters, particularly the calcifiers that use biomineralization process for forming their shells. So for those who don't know what is ocean acidification, this is like a quick intro. Most of you might be knowing already what is ocean acidification, but for those who don't know, so humans, we emit excessive amount of carbon dioxide into the atmosphere, and this carbon dioxide gets absorbed into the ocean, and this reacts with the seawater, and it produces lots of hydrogen ions. This increase in hydrogen ion leads to decrease in pH, which is called as ocean acidification. So acidification here is a relative term. Usually the ocean pH is around 8.1, and when there is acidification, the pH gets reduced to 7.9 or 7.8. It might look like a very small change, but it's actually a huge change for these organisms. They require a particular pH for efficiently making their shells. So this reduction in the pH or acidification has negative effect over shell formation, not only in oyster species, but also in other foundation species such as corals and mussels, etc. So the ocean acidification process is is not something that's going to happen in future. It's already happening. You can see from this picture that there is drastic decrease in the pH of the ocean in the past few decades. And so there are also studies which show that several oyster species suffer because of this ocean acidification. So this picture shows larvae of Pacific oyster. Under control conditions, you can see the larvae is perfect. It forms D-shaped shell. The larvae is growing well, fine. But under ocean acidification scenario, you can see the larvae is struggling to form its shell. It's like dissolving. So this is a problem. And my research is particularly about Hong Kong oysters because different species they have different sensitivity for ocean acidification. So my project aim was to look at how Hong Kong oysters perform under ocean acidification, particularly their shell formation. To do this, we bring in oysters from the field to laboratory to mimic laboratory condition to mimic field conditions. We mimic control condition, which is, as I said, pH 8.1, like the normal pH condition that is suitable for this organism is the control. And for ocean acidification condition, we bubble carbon dioxide into the seawater, and we make conditions that are known to happen in the future. For example, there are projections that says how much pH will go down in the year 2100 or 2300. So we use those values for we simulate those conditions in the laboratory for the ocean acidification treatment. So we grow the oysters under these two conditions in the laboratory, and then we observe how these oysters perform. And surprisingly, Hong Kong oysters actually they grow their shell uh, normally under ocean acidification and also control. In several studies, several oyster species they struggle to form their shells. Their growth rate is less. Their shell growth is affected under ocean acidification. But however, Hong Kong oysters their shell length and shell growth rate was normal. However, they were severely dissolving under ocean acidification. As you can see, under control condition, the top layer, which is the organic layer, it remains intact. But however, under ocean acidification, you can see the shell is starting to dissolve severely. And then we looked at the hardness of these shells, how hard these shells are. There was no significant difference between the control and the ocean acidification but conditions, but this was a short-term experiment over a few months. So when if you continue growing them under ocean acidification, potentially there could be some mechanical properties affected due to this dissolution because they are severely dissolving under ocean acidification. And also, this picture shows juvenile stage after they settle down. The spat. This is not the free swimming stage, right? This is the early juvenile stage. And when we look at the larval stage as well, we just look at the light microscope. It seems like they are doing fine. The larvae are doing fine. But however, when we look under electron microscope, we can also see there is dissolution in larval stages as well, starting from their hinge region. They slowly start to dissolve. So the dissolution starts as early as in the larval stages. So the conclusion is in Hong Kong oysters, ocean acidification is more of a dissolution problem because they were able to form their shells. They were still strong, but even this resilient species has a problem of dissolution under ocean acidification. We also do several other studies for understanding how the species is able to survive under ocean acidification. We also do gene expression studies and DNA methylation studies, but I'm not sharing those results here to make it simple. This is what we identify. Hong Kong oysters, they can perform a little better than other species under ocean acidification, which is the good news actually. And then with this, I come to the next part, Hong Kong's first oyster hatchery. So first, why do we need an oyster hatchery? Because Hong Kong oyster farmers depend mainly on natural seeds available in the environment, or they import seeds from mainland China. For so they import seeds from mainland, like just after the larvae settle down, they buy as small spats, and then they grow them in the ocean for a few months, 6 months, or 1 year, and then after they become marketable size, they sell it in the market. But we don't have our own oyster hatchery in Hong Kong, even though the industry has a cultural heritage of over 700 years. There is no oyster hatchery, and also there is less natural seed recruitment in this area due to environmental stress and also lots of pollution. So there is a need for hatchery to produce oyster seeds. So we can bring in hatchery. What we do is we bring in adult oysters, we make them release eggs and sperms, we fertilize them, we grow them until they settle down. We grow the entire free swimming stage in the hatchery. And then, after once they settle down as spats, we give them to the farmer. This is the idea of hatchery. Also, another reason why we need hatchery is, you can see here there is string of oysters. So in a shell, in an empty shell, we make the larvae settle down, and they settle down and grow as spats. So at this stage we give it to the farmers, so they grow in the field in the rafts as cluster of oysters. But usually when oysters are grown as clusters, they don't grow much bigger, and also they have very less market value. So instead, when you make single oysters instead of clusters, when you have single oysters, they grow much bigger, and they can also be sold for much higher market value. So the farmers can be self-sufficient with the hatchery that we have here in Hong Kong. They need not import seeds from mainland or elsewhere, and also they can have single oyster culture, which will be a new technique for them, and they can increase their income because slowly these days, oyster aquaculture industry in Hong Kong is decreasing. More and more young people don't want to do aquaculture. So when we introduce these modern techniques, we believe we can encourage more people to participate in this aquaculture industry, and we hope they get better income. And for doing the single oysters, we also have these two new systems. This is a prototype. We are currently, the hatchery is still not constructed completely. We are still building the hatchery. So this system is for keeping the larvae suspended. So the system is the flow through system. So we put the larvae in these systems together with tiny grain of sand like particles when they are about to settle down. So they are floating together with the sand, and they can settle down on these sand like particles. So they can be single oysters instead of settling on a shell or a surface and get clustered. So they will be single oysters. So this is the idea behind, and we are collaborating with our university architecture department for making these models, and we hope this helps the oyster farmers to improve their business, and also we can save the local tradition and also aquaculture. So with this, I finished about two things today. I talked about how ocean acidification is affecting Hong Kong oysters, not only just Hong Kong oysters, several oyster species, and also other calcifiers, and how hatcheries play an important role in sustainable seed supply for the region. So with this, I would like to thank my lab members and also my husband Santosh, who has been a huge support during my research journey. And also, I am currently 32 weeks pregnant, and I would like to thank my baby for being patient during my research and all my talk and everything. So I also would like to thank the organizers. Thanks for listening to my talk. Thank you.
All right. Well, first of all, congratulations.
Mhm.
That's pretty darn exciting. And yeah, it is very interesting research, and it has all kinds of implications with the way that our oceans are changing right now from the CO2 that we are putting into the atmosphere. So yeah, very important research. Now, before we ask a couple questions, I just want to ask a couple questions about the oysters in general. One being, how long does it take for them to go from that larval stage to, that larger shelled stage?
Usually, it depends on the species as well and how much you feed them. If
Mhm.
you feed them more, they can grow faster and reach the metamorphosis stage much earlier. But approximately, it takes at least 15 days, 10 to 15 days for them to uh, settle down as pats.
Yeah.
And after that, a maximum they can take is like a month if there's not enough nutrition. So, from our experience in the hatchery, if we feed them more, sometimes they can grow faster and reach the settlement stage much earlier. Yeah.
Okay, that's faster than I was expecting. All right.
Mhm.
What about lifespan in general? How long can an oyster? What's an average lifespan for an oyster? Maybe the
We
Hong
have
Kong one.
seen even like 12, 13 years old long oyster shells. But typically for aquaculture, they just fatten it for 1 year or 2 years and then they harvest. Yeah.
Yeah. So when in the wild, when one's living like up to 10 to 12 years, um, are they constantly growing, increasing in
Yeah,
size?
yeah, yeah. You can see their shell size is much bigger, especially Hong Kong oysters. They grow like really long instead of like
Okay.
this. They grow like really long when they are in the, uh, so you can see really long shells. Yeah.
Yeah. And then are there growth rings, maybe similar to a tree? Is there
Yeah,
somewhat?
you can see like several layers, and with that also you can tell
Okay. So I'm curious about just in general, what's the Hong Kong population of oysters like in the wild?
what do you mean, like species?
Yeah. Yeah.
So in Hong Kong, there are several species, but the one we focus on is Crustaceea hongkongensis, which is
Huh?
found in the Deep Bay area. But these days, oyster farmers, uh, even though our experiments show this is like one parameter, right, ocean acidification,
Yep.
but in the field, sometimes in summer there is huge mortality, in winter there's huge mortality. It could be due to pollution. We have a project ongoing in our lab for figuring out those. So sometimes oyster farmers, they think, oh, this species is not performing well, so they will buy some other species
Right.
because they're just importing the seeds, right? So they will, for aquaculture, they mainly use Hong Kongensis and also Angulata, Crustaceea angulata. Yeah,
Okay.
sometimes. Yeah.
Well, it sounds like I guess there's a little bit of good news in the results in that the Hong Kong oysters, they're on maybe the more tougher scale
Mm-hmm.
where the larvae still able to develop a shell. It's still hard. It is dissolving a
Mm-hmm.
bit, but it's still, I guess,
Yeah.
viable. It's still,
But this is just one parameter. But in the real scenario, there will be multiple stressors. So we also plan to run other multiple stressor experiments to look at like how they perform under several stressors together, and
yeah.
to give a solution for that.
That's exactly the question I wanted to ask you next was what was next with your research? So it sounds like you're going to bring multiple stressors in, more like, they're experiencing now. And then what
Mhm.
about, I think you said 8.1 is normal, but you're seeing pH around 7.9. Do you experiment? Because, with more carbon entering the atmosphere, that the pH could lower even more. Do you do tests at lower levels to see what's going to happen?
Yeah. In Hong Kong, this area, like usually this species, they grow. The pH is already like 7.8, 7.9, like average. So, for my experiment, I actually used much lower value, 7.4. So they're still able to survive under these conditions because I think they're also exposed to fluctuating pH in the
Yeah.
region where they go. So it depends on the species. If a species is not has not experienced low pH before, then that species will tend to perform much worse than this particular species, probably. Yeah.
Okay. Well, before we wrap up for today, I do want to share. Oops, I lost my banners. There they are. I do want to share your Instagram at
Thank
Tamil
you.
Scientist, and then your YouTube channel. Tell us a little bit about your YouTube channel.
Oh, I particularly make videos in Tamil language as a South Indian language. I'm from the state Tamil Nadu, so uh, I make videos not just about the ocean or ocean acidification. My background is biotechnology. Actually, I did my bachelor's and master's in biotech. So during the pandemic, I started this channel because there was lots of misinformation going
Yeah.
around. So at that time, I started this channel to raise awareness about like fake news about vaccines and stuff like that. So I talk about a wide range of topics related to biotech and biology, ocean, etc. Yeah,
Yeah. No,
but
it's great.
mainly in Tamil language for the local population because many of them they don't understand English.
Yeah.
So I think it's important to communicate science in local languages as well.
No question. Science communication is so important. If scientists are just communicating with each other,
Mhm.
uh, you're missing the people who need to hear it as well. And so
Yeah.
communicating your science in an easy to understand way, a way
Mhm.
that's relevant to people is so important. So yeah, science communication is really important. And I'd love to see more labs and more expeditions who are realizing that and bringing science communicators into the fold to
Yeah.
help spread the knowledge, the new learning, and what it all means.
Yeah, sure, sure. And you're doing a great job with these festival and also your other work. I really appreciate. I am really it's very inspiring for me as well. For
Well, I appreciate that, but we couldn't do it without people like yourself taking time to share your story, share your work, and really just open our eyes to the biodiversity that's all around us. So, Kanmani, thank you so much for being with us tonight. Thank you for the
yeah.
work you're doing. Good luck with the new edition coming down the line. That's so cool. And again, thank you so much for joining us for the festival.
Thank you so much. So, see you.
All right, have a great rest of the weekend.
You